So we will start. This is the last lecture. So hopefully you will learn about food and waterborne infections. We are going to cover some key foodborne pathogens, reservoirs, sources, routes of transmission of infection, some methods of detection, and also symptoms and prevention strategies. So foodborne illness. So what is a foodborne illness? Human disease and sickness after eating contaminated food with either infectious or toxic agents. So when I say infectious agents, we all know what infectious agents are. These are pathogenic microbes. They can be fungi, they can be bacteria, they can be protozoa. And when I say toxic agent, it means either there are physical agents, which is like there is like stones in rice, for example, or chemical agents, which is nothing but sometimes chemicals are released over time in a particular food substance that enter body through ingestion of food or water. So broadly speaking, the disease is classified under food and waterborne diseases. It is not just foodborne diseases, but water associated diseases are also classed under the same umbrella. On the contrary to that, we have got food spoilage, which is undesirable change in the appearance, texture, odor, and flavor of food, making it unacceptable for human consumption, but not necessarily harmful. So you can have food that can be, it is unacceptable for consumption, but not necessarily it is harmful. What is the example of that? Over there are two things that are listed. One is a physical change and another one is a chemical change. In physical change, there is loss of moisture. Do you know, or can anyone give me an example of a physical change that they have seen in a food? No, that is not a physical change. Physical meaning physical. Mold is an additional thing over there. What is a physical change? How many of you all had the uh, what? Dum aloo kosha na yesterday? How many of you all had that? You all had, right? Sometimes at home we have potatoes, right? And what happens to the potatoes? They shrink sometimes. I'm sure everyone has seen that. So that is kind of a physical change. There is a loss of moisture over there. Okay, you can also have bread, for example. You have your toasted bread, or you have bread that you have left for some time. You might not necessarily have mold on it, right? But it will start getting, um, it will start losing moisture from it, basically. So that is again physical change. And then chemical change, we have got oxidation. Sometimes you have got chemicals produced like solanin, which I'm sure again people might have seen. So when the potatoes are turning green, so that is solanin production. Uh, And oxidation, anyone here knows about oxidation? Yes, apple turning brown, or you have got, your aubergine or brinjal or eggplant, whatever you call it, when you cut those and you are saying that, brown, black color. So that is oxidation. So that is again chemical change. Okay. So foodborne disease causing microbes. In bacteria, we have got some examples. Vibrio cholerae is a common one that causes cholera. Salmonella enterica, um, you know, Serovar typhi, paratyphi A, paratyphi B, C. Those are the ones that cause typhoid. Campylobacter is a very common one, especially in the UK and US, also in like Australia, New Zealand. Um, so that is the one that causes campylobacteriosis. It is also prevalent in African countries as well. It is one of the four main causes of bacterial gastrointestinal disease in the world, according to the World Health Organization. So it is an important pathogen. And I think you know again that I have worked on Campylobacter, or I work quite a lot on Campylobacter. It is associated with stunting in children as well, this particular bacterium. It is these are new studies, so there can be people who are carriers of Campylobacter but not necessarily uh, showcase the symptoms associated with campylobacteriosis. In viruses, we have got hepatitis A virus, that is a food and waterborne pathogen. Norovirus is very, very common again in the United Kingdom uh, and the West. Norovirus is also very common on cruise ships. If you all go on cruise ships, then you will see that there can be an outbreak of norovirus. So it is a winter vomiting disease, or Norwalk-like virus is what it is called, and it is a major foodborne pathogen. It is very common in the winter because during the winter time, um, you know, people are enclosed in the house or on a cruise ship. That is an environment where people are all gathered together, and that gathering or that uh, crowded environment is a ground for these type of pathogens to thrive and survive. Seafood also is associated with transmission of, norovirus. So that is another thing to bear in mind. Then we have got like prions, which are protein-like particles, and bovine spongiform encephalopathy or Creutzfeldt-Jacobs disease is something that is caused by prions. Scrapie is a disease in sheep that is caused by prions as well. In fungi, we have got Aspergillus flavus that produces what is known as aflatoxin, and Penicillium verrucosum that produces ochratoxins. And then lastly, we have got the protozoan parasites, Giardia, that causes giardiasis, a type of diarrheal disease. And we have also got Cryptosporidium parvum that causes cryptosporidiosis. So burden of infectious diseases. So burden is measured by combining two indicators: the number of years lived with disability as a result of disease and the number of years of life lost to disease. So this is again very, very common measure. And in global burden of diseases, whenever we are calculating the global burden of diseases, or when we are analyzing graphs and other things with international experts, we look at these parameters: DALY, disability adjusted life years. So something to bear in mind or something to remember. It is a very, very common indicator, DALY. DALY is nothing but YLD, years of life with disease, plus YLL, years of life lost. Okay, so that is the indication of disability adjusted life years. And I think we know that when we are having disability, the life years will, decrease. So what it tells you is you are supposed to live for maybe, I don't know, 95 years, but because of the disability and other things, the adjusted life years are less. So you are going to live, I don't know, like 60 years or something like that, depending on the types of diseases that, or the types of underlying conditions, and the risk factors that are there along with it, environmental exposure and other things that impact human health. So sources of food poisoning, I don't need to tell you all what sources are there, but over here you can clearly see poultry meat um, was the food linked to most cases of food poisoning. Uh, in the United Kingdom, we have around 750,000 cases each year. There is also 10% underreporting. We also have, produce that includes vegetables, fruits, nuts, um, causing the second highest number of illnesses. And then lastly, we have got the beef and lamb that is responsible for uh, food poisoning cases. So this is just a report of, a restaurant that has got like a 0-star rating. In the United Kingdom, we have what is known as the Food Standards Agency, or FSA, and that agency is the one that inspects premises, and it gives rating from 1 up to 5 stars. So if you fail the rating, then basically you might have to do major improvements, or you might have to close the restaurant. That is the option over there. So you can see over here that this is a restaurant that was issued a 0-star rating. And 0-star rating meaning, I don't need to tell you all what unhygiene looks like. Everyone here knows. It can be anything from pests to not washing things correctly to, cross contamination of things, I mean, no, yeah, I'm. So common GI infections in England and Wales, you can see again very commonly reported cases here. You can see Campylobacter, of course, which is the major pathogen, but other than that, you have Cryptosporidium, Giardia, Salmonella, Shigella, S-Tech E. coli, or S-Tech 0157 is nothing but E. coli 0157, or Shiga toxin producing E. coli. S-Tech is also called as V-Tech, virocytotoxin producing E. coli. Then you have got rotavirus and norovirus as well. Rotavirus was a major pathogen here in India, but there is a lot of positive drive towards eliminating rotavirus, and there are vaccines as well here in India, and we have been very lucky to decrease the cases of rotavirus, which is a positive step. So you can see that over here, like the 9 out of 20, 10 out of 20, that is nothing but weak number. And then the year, what is the year? And then total reports are highlighted, then cumulative total is highlighted. And then it is compared to the previous years. It is compared on a yearly basis as well, which diseases are rising, which diseases are decreasing. So routes of transmission, definitely human to human transmission is very, very common. Poor sanitation is a major factor, you know. So that's why hand hygiene is very important, making sure that you consciously clean your hands before you eat food or anything else. Zoonotic transmission is nothing but transmission, between animals and humans via fecal-oral route. As I said to you earlier, 6 out of 10 and 3 out of 5 emerging infectious diseases directly have their origins in animals. So animals is a major source, and zoonosis is nothing but it is the transmission from animals to humans. And then we have also got environmental sources where you can see you have got the soil, you have also got the water. water streams that are contaminated, or the soil that is contaminated with feces of ruminants like cows or sheep, um, you know, or there is a, water runoff associated, linked with that to the neighboring rivers, and maybe those neighboring rivers are a source of tap water in the rural areas or in the rural regions. So these things are something that we need to consider, because we live on the urban side. It is not like, oh, uh, we should not be thinking, because in the rural side, we still have a place where I come from. We still have got our bore wells, for example. So understanding that is important. What it is exposed to, what is there in the neighboring regions. Is there a sewage treatment, or is there a septic tank or something that is close? Because you believe it or not, when I was a bachelor student, we were testing the potability of water. And when we tested the potability of water, obviously in our water that we had collected from the bore well, there was no contamination as such, because our bore well was in the front of the house, and our bathroom and toilet was right at the end, at another end of the house. But as opposed to that, one of our friends from another neighboring village, she had got water, and I mean, she claimed that they boil water and all that is fine. But her water, I still remember, had the Enterobacter species, Enterobacter aerogenes that we discussed in the previous lectures. So it had got Enterobacter also, and it is from that only. It is from the septic tank or whatever that is nearby. So these things are something that we need to consider. It is not like, obviously, I have got the experience of this, because I have lots of real life incidences. Almost for each and every disease I can tell you in my life who was affected. Literally, yes. Whether it is someone in the United Kingdom, or someone in India, or someone elsewhere. And that is something that allows you to understand the topic very deeply as well, because it is personally associated with you. So anyways, so exotoxins are nothing but proteins, so produced by gram-positive bacteria, released outside the cell, because bacteria can grow and proliferate. So as the name is suggesting, exo, outside. So these are non-pyrogenic. Okay, that this is something you have to know or remember. Non-pyrogenic meaning, non meaning no, and pyrogenic meaning, pyros means heat and genis means to give rise. Everyone has got fever sometime in their life. So fever is nothing but it is pyrogenic. So non-pyrogenic meaning not fever causing. So some gram-negative bacteria can also produce exotoxins, but primarily gram-positives, not heat stable. Okay. So when I say not heat stable, it means that the toxin that is produced, that is heat labile. Okay. So 60 to 80 degrees centigrade, it will get denatured. But the toxin is highly toxic, which means that microgram of that toxin can, kill a person. It is highly fatal. Example of that is, some examples are botulinum toxin produced by Clostridioides botulinum, and tetanus toxin produced by Clostridioides tetani. Then we have got the endotoxin. So endotoxins are nothing but these are lipids. Gram-negative bacteria are obviously, are the ones that have got the lipopolysaccharides. So this is nothing but it is the integral part of the outer membrane of the cell wall. So the outer membrane of the cell wall of the gram negatives, that itself is the endotoxin. Remember that. Okay, there is nothing else. So it is not released until the cell lysis occurs. So cell has to lyse, and then the, you know, reaction happens, and this is pyrogenic, which means that it will give rise to fever. It is heat stable, which means that up to 250 degrees centigrades, it can still be stable. Moderately toxic, but less fatal. And examples of these are some toxins produced by E. coli and then toxin produced by Vibrio species. So next we talk about Bacillus cereus, which is another environmental pathogen associated with soil. But lots of other dried foods like rice, spices, beans are also associated with Bacillus cereus. The illness is self-limiting. If you don't know what is self-limiting, it means that it will just go off after a few days, maybe 5 days or a week. Like cold, we have all got common cold in some part of our lives, and cold is also a self-limiting illness. It will just clear by itself. So in the same way, this is a self-limiting illness. It takes less than 24 hours for the symptoms to manifest, and this is intoxication as opposed to infection. And there are two forms. There are two forms of this illness. One is the emetic form. Okay. So if you don't know what is emetic form, emesis means vomiting. Emesis, the Latin word emesis stands for vomiting. So emetic form is nothing but you will see abdominal cramps, nausea, vomiting. And the toxin produced during that stage is a heat stable toxin. Okay, so it is stable. And the other form is the diarrheal form. So after 6 hours or so, you will see that you will have abdominal cramps and diarrhea, and that toxin is a heat labile toxin. You can see the picture of Bacillus cereus in the gram standard is highlighted on the right side of the image. So this is how it looks like. You have got the B cereus present in dried rice. Spores survive cooking. Spores germinate, grow, and release toxin at room temperature. In the past, I told you there are spores that are present in soil. There are spores that are present in other foodstuffs as well. And when the environment is optimal, when there is nutrients, and over here you can see the rice is nice and fluffy, and it gives a nice environment. So that is the reason why they will germinate. And what I mean by germination is that the round spore that is there, it will come to the actual form of bacillus, like it will start becoming rod. And they will release toxin at room temperature. Sometimes when you are reheating, you will see that the toxins remain, they are not destroyed. People eat that, then in 1 hour of service, you go and you start vomiting. So that is what is going to happen. So there are a lot of precautions that need to be taken, and also, you know, um, we have to be careful what we are eating, how we are eating, how many times we are reheating the rice. Most of you all, if I ask you, maybe you will not even know what the rules and regulations are for reheating of rice and all these other things. So we will see now the prevention strategies, what should be done, and hopefully you can apply that. So ideally, serve rice as soon as it has been cooked in the ideal form. That is what you do. If that is not possible, cool the rice as quickly as possible, ideally within 1 hour. Okay, cool it as keep the rice in fridge for no more than 1 day until reheating. Okay, so that is how much time you should be keeping the rice. When you reheat the rice, always check that it is steaming hot all the way through. And then don't reheat rice more than once. That is the most important thing. Maybe they will tell you at home that, don't reheat the rice, but this is the reason for that, because of the toxins that are produced. And sometimes you feel, no, like when you have eaten this kind of reheated, I'm pretty sure everyone here has got the experience of that. And then the tummy is rumbling. You are having the rumbling sound in your stomach. Maybe you will not go to the far stage of diarrhea, but maybe you will see that uneasiness in your stomach. And that is all because I think I highlighted to you what the prevention strategy looks like. And sometimes we just have the tendency to just ignore, but we should be consciously thinking of um, what to do, and especially those who love rice. I personally don't love rice a lot. My wife loves rice, but we have to follow these strategies. So next we have Clostridioides botulinum. It's a gram-positive rod-shaped bacterium, produces subterminal oval endospores, bulging endospores. You can see maybe in the picture, it will look like a badminton racket. That is a classic example, or that is a classic indication of Clostridioides. Badminton racket shape appearance is what you see. And there are lots of toxins, obviously, produced by Clostridioides botulinum. Uh, Botulinum toxin is the main one, but you can see that there are different forms of botulinum. Type A, most toxic form of human botulism. Um, Type B is less toxic to humans. Type C causes botulism in fowls and cattle. Type D associated with forage poisoning. Forage poisoning is nothing but, when cattle and all these animals are eating things around the farm, and then they come down with uh, this infection, Clostridioides infection. Then we have got from fish and fish products. Type F is human botulism. Type G is not implicated in any disease as such, and type H is again implicated in human infections. So the ones that are highlighted in red, those ones are the important ones for human infection, is something that you should know. So where do we find, soil is a rich source, obviously, of um, Clostridioides infection. Um, Dust, river, or sea sediments. If food is also there, chicken meat, for example, can have spores associated with Clostridioides. Uh, then infant botulism is there. You can also have wounds that can be contaminated with Clostridioides species. Anyone knows what is infant botulism? So why it happens when they are given honey, or who is given a honey? Children of how much age? 1. 1 year of age. Up to 1 year of age, you can't or you should not give honey to the child. And I think everyone knows that. No one knows the reason behind, or people generally not tend to understand the reason. But reason behind that is obviously honey can contain spores of Clostridioides, and if you're giving that to the child, then the child immune system is low. You know, when the child immune system is low, it can come down with the severe form of botulism, and that is why you should not be giving honey to children up to the age of 1. After the age of 1, it is all right, but up to the age of 1, it is a complete no. So more serious and reportable need low salt, neutral pH, anaerobic conditions. So that is another important thing to know. Remember that Clostridoides species, they are anaerobes, which means that strictly no oxygen. They can't grow in presence of oxygen. Germinate and grow and produces potent neurotoxins. Heat labile toxin that is destroyed by cooking, and severe symptoms descending in acute flaccid paralysis. And you can see that in the picture again on your right hand side, which shows limb weakness and paralysis. How some people can experience these symptoms. Home canning, native meat aging, and oils can be implicated again in this. Sometimes we in the United Kingdom have cans of food. Processed food is there in the form of cans, and sometimes you can see cans have not are distorted, or they are bulging or whatever, and as a result of it is result of this sometimes Clostridoides that is contaminated. So next is Salmonella. Mammals are affected. Birds and reptiles are the hosts. Transmitted through contaminated food, raw meat, poultry sometimes, eggs, dairy products, chocolate as well. Live strains of Salmonella enteritidis and Salmonella typhimurium are given as vaccines in the United Kingdom, and that is a result why the numbers, especially in poultry, have decreased, and as a result of that, Salmonella contaminated eggs, the numbers of those are low. So UK is the fourth largest European egg producer with 11.2% of all laying hens, but has the lowest instances of Salmonella in the whole of Europe at 0.1%. So it is very, very low because of the vaccination strategy that has revolutionized that. And you can see in the picture XLD agar that shows Salmonella colonies. So commonly used lab media for isolation of Salmonella, desoxycholate citrate agar is there. Then you have got XLD agar, you have got Salmonella shigella agar, which is a extended form of DCA agar. And we have also got McConkey agar. So in the DCA agar, sodium citrate is there, ferric citrate is there, lactose is there, neutral red is there. All the usual suspects are there. And thiosulfate is there. So as a result of thiosulfate, you will see gas production, hydrogen sulfide gas production. That is why you see the blackening of colonies. So compared to Shigella, Salmonella will produce black colonies, pin black colonies. Shigella doesn't produce black colonies. And then McConkey agar, we all know. Salmonella is a lactose non-fermenter, if I'm not wrong. Yeah. So 35 to 37 degrees Celsius for 24 hours is the incubation period, and then Vidal test is a widely used diagnostic test for salmonella infections for enteric fever. This is antigen antibody reaction. If they are suspecting salmonella infection, then they might be doing Vidal test for you, or for anyone in which they are suspecting. So you can see an egg over here, and you can see a lion mark on this particular egg. Perhaps you have never seen this picture before, but this is a very common mark in the United Kingdom, and it just indicates that the egg from the egg that you have on your plate, it has come from a specific farm. It has got the best before date. It has got the farm ID. It will also highlight the method of farming and also show that those hens are vaccinated against salmonella and produced to a strict code of practice. So if the lion mark is there, it is like voting. You know what is voting, right? When you vote, you get a mark on your finger. So here there is a stamp on egg. If it meets all the standards, and it is also good, right? Because you have traceability of the egg, and that is the reason why we don't have any salmonella cases in the United Kingdom, or the incidence is so low, very, very low. Um, so this is a very good practice, or this is something that is regulated. This is not like individual farm will be doing what they want. No, this is a standard practice. All the lion marks look in the same way, and they are written in the same manner with the um, you know other details that are there. So next we speak about Vibrio. A Vibrio species. Vibrio stands for vibration. If you don't know, that is the name. That is how the name is derived. So gram-negative rod-shaped bacteria, non-sporulating. They don't have spores. Non-capsulated, facultative anaerobes. They can be curved or comma-shaped. So comma is a common form. They are highly motile due to single polar flagellum. There is one flagellum at one end. When cultured on agar, they form two distinct colony morphology, rugose and smooth, due to population diversity and adaptation to the conditions. And you can see those two colonies that are there, one at 37 degrees and another one at 22 degrees. TCBS is the selective medium that is used to isolate Vibrio and other pathogenic Vibrio. It has high alkalinity and components like thiosulfate, sodium citrate, ox bile, sodium cholate, which inhibit gram positives. Sucrose fermentation occurs. The indicator over there is bromothiamol blue. Bromothiamol blue is something that turns yellow under acidic condition and blue under alkaline conditions. And then yeast extract and peptone provides nitrogen, vitamins and amino acids for the growth. This table highlights the different species of Vibrio, and it also links with the source of infection, from where it is linked, seawater, seafood, fresh water, and then the roots of infection, and also highlighting some clinical manifestations. And you can see that Vibrio cholerae O1, O139, again over here, the O stands for the somatic antigen, if you remember how the typing was done in the previous class. Then you have got Vibrio parahemolyticus. Vibrio vulnificus is an important pathogen linked with like seafood. You can see that it is present in seafood. Seawater can cause gastroenteritis, wound infections, and sepsis. And there are other types of vibrio as well that can cause wound, eye, ear infections, and so on. So Campylobacter species, I already told you a little bit about Campylobacter, but this is a microaerophilic organism. It likes low oxygen. Sources are mostly domestic animals. Major cause or the major reservoir of Campylobacter is poultry or chickens. So that is the major host reservoir. Other than that, you have got ruminants. Those are the secondary sources of human infection. After ruminants, you can have pigs that can be the source of Campylobacter infection. And then finally, wild birds are also implicated in Campylobacteriosis. And the strains that are there, some strains are specific to poultry, some strains are specific to ruminants. So when the strains are host restricted, those strains are called as specialist strains. Okay, but if the strains are moving in between, some strains are there that are found in cattle, some strains are found in poultry, some overlap is there. So some dynamic flow of strains is there between reservoirs, and those strains are called as generalists. Okay. So specialist, I think everyone knows what is a specialist, right? It is only one, and generalist because it is moving in between. So that is something that you might write down or remember. So fecal-oral route is the common route. Foul smelling, bloody diarrhea. You can have bloody diarrhea. Incubation period is 2 to 5 days. Excuse me. Commonest cause of bacterial diarrhea, food poisoning in the UK as well as in other Western countries. You can see the colonies of Campylobacter on the agar that is highlighted on the right hand side, bottom right hand side, and on top you can see the spiral shape, uh, staining gram negative cells of Campylobacter. So Campylos means curved and bacter means rod. So that is how the name is derived. It is microaerophilic. So as I said to you, it doesn't like oxygen. So only 5% oxygen, 10% carbon dioxide, 85% nitrogen. Okay, and 37 degrees Celsius for 48 hours is the normal incubation period. So plates are incubated in a chamber that has these gases. So we have got the microaerophilic Don-Whitley chambers, they are called, for either for anaerobes or microaerobes. And then you have gas that is pumped from one end. You also have gas, you also have jars. You can also put the plates in certain type of jars and you put a sachet of packet in those jars, and there's a reaction with the oxygen in the jar and microaerophilic conditions are created over there. So that is another, that is a method in the low and resource limited setting where they will not have access to these kind of gas cylinders that have this composition. So, but it is not an easy organism to grow. It is a difficult organism to grow. You can see nice shiny silver colonies on the charcoal agar, modified charcoal cefperazone deoxycholate agar, CCDA. So it is a selective medium for isolation of Campylobacter species from a variety of samples. Sometimes we also do what is known as enrichment. We have the sample and we enrich the sample in certain media because the viable number of those organisms are very low. And you can apply that enrichment in your own studies as well. For example, if you're not getting any isolates or you're finding it very difficult, then you should be considering the enrichment procedure because not everyone thinks like that, but I'm giving you some suggestions along the way to consider. Because once you enrich, then maybe you will find something of relevance over there. And enrichment is done using like double strength, triple strength, broth or whatever. Sometimes we have like blood and other kind of nutrients which is the enriched source of nutrients. Lysed blood is a very common enriched source of nutrients. So it is gram-negative curve-shaped bacterium. As I highlighted earlier, one of the 4 key global cause of diarrheal disease. 5 days incubation period, 750,000 cases per year. Cases per 100,000 of the population is nothing but incidence. This is a term that is used very common in epidemiology. Cases per 100,000 of a population. Remember that or write it down. It is universal term, whether you are a virologist, whether you are a parasitologist, whether you are, I don't know, whatever, mycologist. When you are doing epidemiology and other things, you will always report cases per 100,000. Not cases per 1,000, not cases per, 10,000. No, only per 100,000. So if you have population of 1 million, you have population of 1 crore or whatever, you always calculate it according to that. Roughly around 200 to 50 deaths is what it causes each year in the United Kingdom. So you can see that it is not a pathogen that leads to many deaths, but economic burden is very, very high. It costs the UK economy around £1 billion each year in the time lost from work. For example, people are not going to work, and then that has an impact on the economy. If you're a software engineer and you're involved in a project, in 5 days you didn't go to work. So, it has an impact on the economy. So that is the impact. Up to £1 billion each year is what it will lead to. Or a child is ill with campylobacteriosis, and then, you need to have your parents at home. Self-limiting infection requires electrolytes and replacement fluid. I think everyone knows what electrolytes are. Oral rehydration fluid is a common example. Common symptoms include abdominal pain, diarrhea, vomiting, fever, nausea, blood in feces. Campylobacteriosis can affect immunocompromised patients as well. So elderly and those that are young. So drug of choice sometimes can be macrolides or fluoroquinolones. Sometimes carbapenems are also given if there is a bloodstream infection suspected. Rarely Campylobacter will cause bloodstream infection, but yes, there are cases that show that it can lead to bloodstream infection, and you might have to give meropenem, carbapenem class of antibiotic. Guillain-Barré syndrome is a very rare, but serious condition that affects nerves and it is a neuromuscular disorder. And again, it is an autoimmune disease that develops. It mainly affects the feet, the hand and limbs, causing problems like numbness, weakness, and pain. So it is a long-term condition. This is something that can't go. And if a person has Guillain-Barré syndrome, then it is again, not a very nice thing. 80,000 people are affected by these type of illness, so it is not a small number. So Guillain-Barré syndrome is also caused by other viruses, but Campylobacter again is a very common organism that will manifest this kind of condition in extreme circumstances. So AMR associated things, again in the previous class when you were doing the phylogenetic tree, you were doing, OXA61 and, oxacillinases. Those were oxacillinases of Campylobacter that you were looking at. So OXA61 like gene is the very common gene that is present in Campylobacter. OXA184 is also a very common one. Then 23S rRNA gene mutations can confer resistance to macrolides. We have got TET-O like genes. So some TET-O like mosaic genes are also there. Mosaic gene is nothing but, two or three tetracycline resistant genes that are combined with each other to form like everyone knows what is a mosaic, right? Mosaic is a pattern that is mixed pattern. And if you don't know what is a mosaic, then I think everyone here has seen a kaleidoscope, right, with the eyes. So over there you see different patterns that is a mosaic. I used to love that when I was a child, but it has been many, many years I have not seen that. Then gyrate mutations, everyone knows the gyrate gene, the topoisomerase 2 gene, yeah. So mutation in that gene will lead to fluoroquinolone resistance. And then there are all sorts of aminoglycoside modifying enzymes that are highlighted. Lincosamide nucleotidyl transferase, then chloramphenicol acetyl transferase. So this is nothing but it is showing the incidence of campylobacteriosis in the United Kingdom over time. And you can see the blue line, which is Scotland, and you can see the red line, which is England and Wales. And you can see the incidence is going up and down. But in the earlier years, the incidence was very low, and now it is gradually increasing. Um, in between, you see the studies that are there, like CAMS, ICAMS 2, ICAMS 3 and 4. Those are the studies that we did in those years. Um, and this is the largest epidemiological study of Campylobacter in the world or in the universe, where we sequence 7,000 or more than 7,000 Campylobacter strains. And I think many of you all know, as a result of that, I also discovered new species of Campylobacter. New species of Acinetobacter were also discovered in that process by mistake, but I was able to identify it as a new species. So incidence is highlighted over here and the calculation is shown. I'm not going to go through the calculation, but you can see very well, incidence, as I said, is always calculated cases per 100,000. And, it is saying like calculate the incidence if there were 6,963 cases of Campylobacter in country A in the year 2023. And the population of country A is highlighted over there, 5,540,000. You just do a cross multiplication and incidence is that number. Okay. So those many cases are there per 100,000 of the population. You just do cross multiplication and you have your answer. Next, we talk about different types of E. coli. We have got the enterohemorrhagic E. coli that can cause HUS, hemolytic uremic syndrome. So this is also called as Shiga toxin producing E. coli or virocytotoxin producing E. coli. And hemolytic uremic syndrome is a severe condition, that can lead to death or kidney failure. General support is by intravenous fluids. Corticosteroids are also advised. Plasma filtration or exchange can be performed. Dialysis is performed, but no antibiotics are given. Why no antibiotics are given? No, that is not the reason. Anyone? I will give you all time to think. I'm not going to give you answer very easily. You take your time to think and you tell me why antibiotics will not be given and what we are studying in this particular presentation. I'm giving you the clue. In this presentation, you are studying something that is linked with this particular question. And if you're able to draw that link, then you will be able to tell me why you will not give antibiotics. Yeah, it will heal, but why you should not give antibiotic? That is the question. Anyways, I'm not going to waste time. We can talk during the break. Next one is enterotoxigenic E. coli. Okay, so first week of travel, traveler's diarrhea is what it causes. Self-limiting, you give oral rehydration solution and you will be fine. Next one is enteroinvasive E. coli. It invades the epithelium, um, involves the large intestine, spread associated with hamburger meat and unpasteurized milk. Enteropathogenic E. coli is also there that affects infants and is common in nurseries and spreads to contaminated water, raw beef and chicken, fecal contamination. Anyone knows now why we don't give antibiotics? What is there in this slide that can give you another clue? Yes. So enterotoxigenic, no? It's virocytotoxigenic, Shiga toxigenic. So that is a toxin. Yeah, after you are giving the antibiotic, you will burst open that cell. Once you're bursting open the cell, a person will die. Hemolytic uremic syndrome is a manifestation of that, and kidney failure occurs as a result of that. So good, well done to you. You have given a good answer. So next is Cryptosporidia. Protozoan parasite mainly affects, which infects mainly animals, including humans. Main host is cattle. This is a parasite, causes diarrhea in young livestock and human illness from, drinking infected water, swimming recreational water, and so on. Chlorination is ineffective, so everyone knows chlorination is done for water to kill microbes, but you can't kill cryptosporidia with chlorination. So filtration is involved with UV or ozone. Contaminated drinking water outbreak associated with this was seen in the United States, where more than 400,000 people were affected, and it led to more than 100 deaths. And then lastly, we have got the hepatitis A, which is the HAV virus spread via fecal-oral route. Patient has unrecognized hepatitis, fecally incontinent, and other risk factors are there, such as eating or drinking in patient care areas, not washing hands after handling infected infant, for example, sharing food, beverages, cigarettes with patients, their families or staff members. So that these are the other risk factors. So that's it for this lecture. We'll quickly do the quiz now.